Morning, everybody. Is that a late breakfast or an early lunch, Robert?
It's a snack.
Oh, fair enough. Keep
I
your
need some
energy
brain
level.
food. Yes, sir. Yeah.
Okay, so some coffee. Okay, we've got a bunch of stuff to do today. So this is a pretty solid PowerPoint. This is the one on translation, well, RNA processing and translation. And so the first part of this is going to be eukaryotic specific, because prokaryotes do not process their RNA. They can actually translate it as soon as it's produced, which we'll get to later. And then once we get through this, we'll then start on translation, which is it's not technically the final part of gene expression, because there's a bunch of stuff that goes on after that, which we'll talk about in the next section. But for our purposes in this section, that's like the last half or third of gene expression. And then right at the very end, way down here, it's a monster of a PowerPoint. We'll look a little bit at the genetic code and the consequences of that. Okay. So, all right, so what we've covered so far, and actually if anyone has any questions about transcription and transcription initiation, now's a good time to ask. But essentially what we've done so far is we've really looked at how the information in DNA is turned into a form that can be used, right? And that form is messenger RNA or mRNA. And so there's obviously a considerable amount of kind of control really that goes into the control, into when, where, and how much a gene is transcribed. But once it's transcribed, obviously that mRNA doesn't do anything in and of itself. I mean, some RNAs do, right? There are RNAs that do stuff, quite a lot of them actually. But for what we're talking about now, we're just focusing on the coding RNA, so mRNAs. So we need to do something with it, right? And so in prokaryotes, it's pretty straightforward. You go straight into translation because there is no nucleus, right? The genetic material, even though it's in kind of one part of the bacterial cell, is still in the cytoplasm, right? And so as soon as that gene is transcribed, that mRNA is available for transcription. Now in eukaryotes, that's not the case. So in eukaryotes, that mRNA that's being produced is actually a precursor mRNA. So we need to do two things to eukaryotic mRNA before it is available for translation. One, we need to process it, right? Because it's in a form that cannot be used. Here's a question for the class. What, at the very least, do we need to remove from a precursor mRNA, a eukaryotic mRNA that's just been transcribed, before we can translate it? What part of that sequence or parts, because there's typically more than one, has to be removed before eukaryotic mRNA can be translated? Think about the structure of a eukaryotic gene. Yeah, good job, Maria. No, hopefully not a mutation, Adam. That would be pretty terrible. No, it's the introns, right? So at the very least, we need to remove the introns for that mRNA to be translated. So that's one thing. I mean, there is other stuff to processing, which we'll go through in a little bit. Second thing is, look where transcription occurs, right? It's in the nucleus. Where does translation occur? In the cytoplasm, or, I mean, rough endoplasmic reticulum too, but that's also in the cytoplasm as well, right? So we have to get that mRNA out from the nucleus into the cytoplasm before it's available for translation. And so the mRNA has to be exported. So two things have to happen to a eukaryotic precursor mRNA. Yep, give me a second, Adam, I'll type this up too. Has to be processed, which is remove introns, add cap and tail, which again we'll get to in a little bit. Second, has to be exported to the cytoplasm where translation occurs. And that's either in the cytoplasm or in the rough, I hate typing this out, plasmic reticulum. Try doing that when you're drunk. Depending on where that protein is going to go, right? So the final destination of the protein determines whether it's translated in the cytoplasm or in the rough endoplasmic reticulum, right? So that's like an added wrinkle further on, which we're not going to worry about right now. And so if you look at this diagram of the nuclear membrane, you can actually see it has holes in it, right? These are pores. They're actually made of proteins, and their job is to find that mRNA. And I'm actually not entirely sure how they do that. Something I probably need to read up on. And grab it and thread it through the nuclear membrane so it pops out into the cytoplasm. And the reverse happens for proteins, actually. So any proteins that are needed in the nucleus have to go the opposite direction. So there's quite a lot of, magic that happens, also known as like molecular biology, but sometimes it appears like magic. And then once that mRNA is out of the nucleus, then it can be translated by a ribosome or ribosomes. All right, so keep that context in mind as we think about this, right? Because it's not just something that happens in the nucleus. You have to take into your mind the picture of the cell, essentially. Where is this stuff happening? Why does it need to get from here to here? What happens once it's here? That kind of stuff. So first, we're going to start with RNA processing, because obviously that's like the first thing that happens. And again, this is for eukaryotes. Sorry for the shouting, but it's important. And this is actually one of many regions, and we've already covered some in transcription, where eukaryotes differ from prokaryotes. And there'll be more when we get to translation. So the aim is to kind of teach you the general basics, but then also highlight how that differs between prokaryotes and eukaryotes. Okay, so one thing that's really useful to bear in mind is that mRNA is very fragile. RNA in general is. I look at it funny and it will just fall to pieces. You have RNases, enzymes that will digest RNA all over your fingers, right? From bacteria that are on your skin, right? You leave DNA out on the bench top for a while. It's like, oh well, that'll be fine. Don't do it again, but it'll be fine. Leave RNA out on the bench top for half an hour, and then when you look at it again, it's gone. Right? It is a very, very fragile molecule. Right? That's why when you look at, some frozen dude or dudette like dug up out of a glacier in Switzerland, you can get DNA out of them fairly readily. Like even though they're like Ötzi the Iceman, Captain America. Yeah, well, he didn't have any DNA taken out of him. He was kind of alive, but and he came back. But Ötzi the Iceman is a cool name actually. A little bit of a distraction. Ötzi the Iceman. He was actually spat out from a glacier in Austria, I think. And it's really cool because they could figure out what he ate for his last meal. I can't actually remember what he ate, but they figured it out. You can look it up. And that he'd also been shot in the back with an arrow, which is pretty rude, right? I don't know how that happened on top of a glacier, but it did 4,000 years ago. But anyway, you can get the DNA out of Ötzi quite fine. You're not going to find any RNA, right? So RNA is, and actually read up on Ötzi the Iceman. It's a cool story. RNA is very fragile, right? And so one area that makes it fragile are the ends, right? So there are RNases that cut in the middle, endonucleases, right? But there are lots that cut from the ends. And so what those enzymes are looking for are bare ends of RNA. Whether it's 5' end or 3' end, that's what they're looking for. And so in terms of processing, what we need to do with eukaryotic mRNA is protect those ends, right? And so the very first end that gets produced is obviously the 5' end, right? And so that gets protected by what's called a 7-methylguanosine cap, or 5' cap, 5' cap there, prime for some reason, prime. And so that 5' cap is not recognized by exonucleases, by RNases. It doesn't look like RNA because it's kind of not. It's a weird, funky kind of structure, which I'll show you in a second. And so as soon as that pre-mRNA is produced, and literally only 20 to 40 nucleotides, just enough for it to emerge from RNA polymerase. Or if you think about how big RNA polymerase is, right, it occupies at least 20 to 40 bases of the DNA sequence at a time. Right, so as soon as that initial bit of mRNA makes its way out of the RNA polymerase, it gets capped with that 7-methylguanosine cap, which looks like, where are we? Here we are, this. So it's really weird looking structure, to be honest, right? So down here, you will have the 5' end of the mRNA. So essentially kind of attached to that last phosphate down here will be the first sugar and base of the transcript. Which is essentially what's shown here in green at the top. Right, imagine that's the transcript as it continues. And so as you can see, this cap is a weird structure consisting of a bunch of kind of look a little bit like nucleotides, but they've got methyl groups on and they're kind of joined funkily by the phosphates, right? So it doesn't look like RNA, even though it kind of is. And this cap structure on the very 5' end of the transcript, it serves two functions. The 5, ooh, is a 7. I call it the MPG, which is short for 7 methyl guanosine cap, right? Serve two functions. One is to protect the end from RNases, which is good. There's no point transcribing a gene if it then just gets immediately chopped up by an RNase. And then the second role is to recruit proteins that help initiation of translation. And also, now we're going to start over the course of this day, right? These 2 lectures, we'll start fleshing out why a eukaryotic gene has the structure that it does. Not completely. We're not going to go into introns in crazy depth, like the purpose of those. We'll look at that when we look at the control of gene expression. But we'll start to look at why we have a 5' UTR, why there's a 3' UTR. You know, all the other different kind of bells and whistles. So hopefully we'll start to make a bit more sense why it's structured the way it is. Anybody have some questions, please just stop me and ask. I'm more than happy to take a break and focus on something. Actually, let's have a look, see if we can find a picture of Ötzi the Iceman, just as a little break. Oh, I spelled it wrong. Spelled it phonetically. There we go, images. There he is. Presumably it looks a little bit better than that in real life. Not looking at his freshest. And this is what they reckon he actually looked like. Not wearing many clothes considering he was wandering over a glacier, and those are pretty chilly places. But yeah, anyway, it's pretty cool. Yeah, the pictures. There you go. That's what he looked like when he popped out. Which is kind of gross if you think about it, going on a hike in, you know, Austrian mountains, yodeling and stuff like that, drinking beer, find some dead body in a river. And there you go, that's him getting shot in the back, wearing more clothes, presumably. They actually found like a nick or a incision in one of his ribs in his back that could only have been caused by an arrow. Don't know whether the person that shot him then kind of grabbed it back or why he got shot. Who knows? Anyway, that's a nice little interlude. Doo-doo. All right, so get back to this bit. Where are we? I was in by my chat box. So also as the transcript is being produced, right? And this is, as with many things, right? These events are not necessarily completely linear. pardon me. And also many things can be happening at the same time. So as the 5' end emerges from the RNA polymerase, it's capped. That has to happen first, and that has to happen quickly. Otherwise, it will start getting chewed up. But then as that's happening, an intron may have been produced, right? And so as those introns become available, and RNA polymerase is basically here, right, where the tip of that arrow is, and it's kind of moving to the right. Again, using the bottom strand as a template, the top strand as a coding strand. And as that continues and those introns are made available, the splicing machinery comes in and cuts them out, not necessarily in the order that they're transcribed. Could be doing it all at once, could be doing the second or third intron first. It doesn't really matter, as long as they're all done before the mRNA is exported to the cytoplasm. And at the very end, so if you remember in eukaryotic transcription, it's terminated by cutting the precursor mRNA. Let's see if I can dig that back out. Here you go. So this is the RAT1 nuclease mediated transcription termination, right? And so after the mRNA is cleaved, right, to allow termination, RNA polymerase keeps on doing its business. This mRNA has to have the 3' end capped, right? And that's what the poly A tail here is all about, right? So that's kind of the overview of RNA processing in eukaryotes. Now we're going to go into the actual details, right? And this is essentially what the transcript will look like once it's been processed. Has a cap, introns removed, poly A tail has been added. Okay, so we've covered adding the cap. Now we need to look at the splicing process, and this is actually a fairly involved. What are you doing, dog? Fairly involved process, right? That requires lots of. Can you go back to the subscan? Sure. So capping. So the 5' capping occurs first as soon as the transcript emerges from the RNA polymerase. Then as introns appear, they're removed, but not necessarily in the order that they transcribed. Because it doesn't really matter what order they're removed in, as long as they're all removed. And after the pre-mRNA has been cleaved by RAT1, poly A tail is added to the 3' end for protection. So the cleavage is for transcription termination, yes. but that essentially free 3' end has to be protected. And that's what the poly A tail is all about. So the two processes work together, basically. That makes sense? Awesome. Which part of the overall process? What do you want me to recap? Oh, you talking about this one? You can just unmute your mic, you know. Might make it easy.
Yeah, so it was actually the one where I forgot. You had touched on it, but it had the different steps.
Oh, you mean in the other PowerPoint?
Yes.
Ah, this one. Is that the one?
Yes.
Yeah, so essentially this is all about, this picture at least focuses on the termination of transcription. And the first step to do so is the precursor mRNA has to be cleaved, right? And so it gets cut some point after, you know, where the 3' UTR would be, right? And so this is really focusing on about how that kind of free end that the RNA polymerase is still transcribing now serves as a substrate for this enzyme RAT1, which basically goes along and terminates transcription. But as a result of that cleavage, now we have a free 3' end here. Oh yeah, this is absolutely and most definitely for eukaryotes. RNA processing only occurs in eukaryotes. I hate typing out eukaryotes. I even spelled it right, that one. And so the 3' capping, the poly A tail, sorry, yeah, I mean, it's kind of a cap, but it's not the same thing. The addition of the poly A tail occurs to this end here, this now free 3' end that has been released, right? So down here, all this part is the mRNA that was just transcribed. And so this 3' end has to be protected, and it's protected by the poly A tail.
Thank you.
Cool. Yeah, so it's really kind of reason why I brought this up is because it kind of ties in another process that you've already learned about with what we're talking about now. So it's kind of nice to pull those things together so it, you know, all starts making a bigger picture, basically. Okay, anyone have any, get back there. Anybody have any other questions? And actually, Rose, that's a kind of an interesting question too, which we'll get to later on and probably in the second lecture today, as to, and even like the next section where we look at control of gene expression, prokaryotes do things quite differently, and that allows them to do other funky stuff, right? Where transcription and translation are very closely linked. In eukaryotes, you can't do that because transcription and translation are very much not linked together, right? Both in time and space. So in eukaryotes, you can make mRNAs here and they get translated here, but also you can make them at one point and they get translated at another time. Actually, so much so that some organisms like coelogens, like I work on, actually puts mRNAs into the oocytes, before basically the eggs of the worm, right? Kind of provisioning them so that when they get fertilized, they have all those mRNAs that can be then translated. You can't do that in prokaryotes because those two things are very closely linked. So that's kind of like a cool thing to add as well. Okay, is everybody ready for splicing? Splicing is full of new words and names and funky stuff. Cool. And it also involves something which is my favorite word, just because it's so ridiculous. And that is snurps. Always kind of slightly, I don't know if any of you have German ancestry. I have a little bit, but I pronounce it kind of like schnapps, which is a peach liqueur. It's very nice. If you have legal age to drink it, obviously. So schnurps, right? Because we're pretending to be a little bit German. So schnurps. And that stands for, and this is always fun to type out, small nuclear ribonucleoproteins. Oh, particles. Yeah, drop that into a conversation party. See how many people still talk with you. Obviously loads, because you'll be super exciting and interesting. That's my experience anyway. Like, yeah, I Smurfs. No. Small blue magical people that get eaten by other things. I can't actually remember all of the characters in much better Fraggle Rock. I used to watch that when I was a kid. If anyone has ever seen that, it's pretty funny. Yeah, so small nuclear ribonucleoprotein particles. There. So I'm going to call them snurps from now on because I don't think we have enough hours in the day for me to say that too many times. Okay, so essentially, RNA processing occurs through the action of a complex of RNA and protein called spliceosomes. Right, and so these small nuclear these small snurps, sorry, there you go, see, I'm not going to say it full again, are basically a mix of protein and RNA molecules. Right, and so they're going to do essentially two things by and large. One is they're going to cut the mRNA because we need to remove those introns, and we're going to stick it back together again, right? So these spliceosomes are pretty wild because they recognize RNA where the introns are, which is no mean feat. They cut the RNA, they bend it round into a knot, and then they stick it back together again. So it's quite an involved process. And at least part of it must have been discovered by Texans, or at least people from the Southwest. In the chat, oh my God, all of that, what, the bit about the Texans or a bit about everything else? Spliceosomes find the intron junctions, which we'll talk about in a second. Cut the mRNA, bend it into a lariat, actually it's a capital L, lariat loop, hence the Texan part, because you know we're all cowboys. Well, I'm not, obviously. I'm about as far from a cowboy as you can get. Anyway, cut it again and stick it all back together. So as you can see, it's a fairly involved process. And also, as a fun kind of side fact, this is also one of the processes that makes use of the 2' OH on RNA. Right, so remember, DNA only has one OH free at the end, the 3' OH. RNA has both a 2' and the 3' OH. Hence RNA, not deoxyribonucleic acid. So this is one of the processes that actually makes use of that in kind of a neat way. Okay, you all ready? It's pretty wild. People catch. I'm kind of mournfully staring at the bottom of my coffee mug, which is sadly empty. There you go. I think that was the last drip. It is sad. There's actually another like cup waiting for me. I can hear my coffee maker ticking. Like, haha, have coffee. So I have to wait for another 23 minutes, which is sad, before I can have another cup. Because obviously I can't leave in the middle of the class. That'd be terrible. Like totally unprofessorial. So first things first, we got to look a little bit at the structure of an intron, right? Yeah, I know, but caffeine, exactly. How I got through my whole undergraduate degree without drinking coffee, I have no idea. So this stuff's really important because before, in, and at the end and after the beginning and end of an intron are a series of bases that are recognized by the spliceosome, by the splicing machinery. And essentially there's kind of three kind of main points. One is the beginning of the intron always has a GU. Remember we were talking about consensus sequences when we're talking about transcription, right? Tata boxes, things like that. We have the same deal here, right? And so always and evermore, you'll always have a GU at the start of the intron and an AG or a CAG, but definitely an AG at the end, always. And you can actually use these sequences to find introns in DNA sequences. That's part of how, you know, when we sequence an organism, we can actually find where the genes are and what they look like and what they might encode in terms of proteins. So we have a 5' splice site. This is where the intron is going to get cut on the 5' end of the intron. And a 3' splice site, this is where it's going to get cut at the other end, right? So we have two short sequences which specify cut here, right? At either end of the intron, the GUAG. Now, these are so important. Like the rest of the intron, other than this branch point, the rest of this intron could just contain whatever, right? Transposons, it can contain like junk, just there, whatever, utterly unimportant. mostly. Obviously, I always have to add that caveat because nothing's ever really that simple. But if these sequences, particularly the GU and the CAG at the end, if those change through mutation, right? You could have as many mutations as you want in the middle, doesn't matter. We're not going to do anything. But if these sequences are mutated, this intron will not be removed. Now think about that in terms of translation, right? The protein coding sequence basically ends here and picks up again here. This bit in the middle shouldn't be translated. So what's going to happen if, say, this G gets changed to a C just by natural random mutation? What's going to happen in terms of translation now? There's actually two different things. One of them is fairly obvious, one of them less so. What will happen if this intron is not removed? What's going to happen to the translated protein? CAG, no, that's a separate issue in DNA replication, Rose. Those repeats are ones. So I'm actually not sure what. Well, let's see. I can tell you because I can't remember the genetic code. Saved my life. CAG. Oh yeah, that's glutamine. That's another polyQ disease. So Rose's question was about Huntington's, and actually ALS is a similar one caused by what's called a polyQ repeat, which is where you have multiple codons for glutamine, which is represented by Q, either CAA or CAG. And so that occurs because DNA polymerase kind of pauses and stutters and repeats those codons, and once they get to a large enough level, they produce proteins that can't fold properly. They form aggregates, and those protein aggregates are what cause the neurodegeneration that you see in ALS and Huntington's that obviously produce those symptoms. This CAG is not translated. It's just a consensus sequence. It isn't a codon. So what would happen if the ribosome continues from exon 1 and starts translating into intron 1? What do you think will happen? Just general ideas. What's going to happen to the protein sequence? Is it going to be what it's supposed to be? You got a 50-50% chance of getting it right. It's either yes or no. So if the no, yay! No, of course not, because this doesn't contain information that's supposed to be translated. So yeah, so essentially the mRNA that will be translated will have extra sequence information, this intron in it, right? So that extra information is going to get translated because a ribosome doesn't know that that intron shouldn't be there. So one thing, at least you'll get a protein with a load of extra, yeah, you'll get a wrong protein produced. Plus, the second exon might be out of frame, and we'll talk about that later. Right. And so that will generally produce a protein which is nonsense, right? It's not going to function properly. Second, right, there are almost certainly stop codons in the same frame in the intron. Because there's no selective pressure to get rid of them, right? Because that intron should not be translated. And so that will end up producing a truncated protein, which is also non-functional. So these, and that's why these splice sites, these nucleotides are so conserved. Because if you get rid of them, that protein produced won't work. And if that protein happens to do something important, as an organism, you're not going to work. Right? So there's a very strong selective pressure against any changes to these nucleotides that are highlighted. The rest of them doesn't matter. But these ones on this picture are incredibly important. And so in addition to those at the ends, which define the splice sites, you also have adenine, which is, you know, a short distance upstream from the 3' splice site. This is where we're going to bend, or actually this is where we're going to attach the 5' end of the intron once we cut it off. So the actual mechanism of this is a little involved, okay? So essentially the first part is, and we're not going to show this is just like the general overview, we're not showing the spliceosome and the different proteins here. The first step is cutting the 5' splice site. Right, so exon 1 and the 5' UTR is separate from the rest of the mRNA. The 5' end of the intron is now bent round and is actually stuck to the 2' OH, or the 2' carbon really, of that adenine, right? That branch point adenine, which you can see in this kind of detailed box here. That forms this, what's called a lariat loop, because it looks like a lasso type thing, I guess. And when that's cut off, now exon 1 and exon 2 are stuck back together again. So that's the overview, right? That's the simplified version. So again, this intron's recognized, 5' end is cut, 5' end of the intron. 5' end of the intron is bent round and stuck back to itself, essentially, at the A, that branch point A, which is just here in the intron. And then the intron is cut off of, pardon me, the exon 2. So it's cut at the 3' splice site. And then exon 1 and exon 2 are stuck back together again to make a continuous piece of mRNA. Okay, any questions so far? Okay. So what does this look like in detail, right? So this is the overview. In detail, well, this is where we get the snurps coming in. Snurps. Oh, what chapters will the test cover? What were those results from again? Above? Oh, so come on, give me a second. Adam, you'd have to look at the reading guide. I don't have it open right now to look at what chapters in the textbook the test will cover. In terms of the PowerPoints, it's going to be from eukaryotic, so it's going to be 12, 13, 15, and 16 in terms of PowerPoint. So the one that we're covering today is going to be the last one that we'll cover before exam 3. No worries. Oh, and so Rose's question, let's get back to so that will be if there are mutations in the splice acceptor site sequences. And so, yeah, these sequences here. So if any of the letters or bases that are in this diagram are changed just by random mutation, you'll either get a nonsense protein, like what Robert was talking about, or you'll get a truncated protein as a result of a stop codon being in frame, right? Which normally wouldn't be there because that intron would be removed. Does that help? Cool. Okay, so this is what happens in detail, and this is where the SNRPs come in. So their job is to do a whole bunch, basically everything, right? Their job is to recognize these splice acceptor sites, right? So for example, and there's U1, 2, 4, and 6 from memory, right? So U1 recognizes the 5' end, U2, cool band, recognizes the, I was a big fan when I was younger, still am kind of, recognizes the branch point, and then essentially all these other ones are involved. What they do is kind of complicated to look at like so. But U1 and these other SNRPs, their job is to cut the 5' end of the intron, right? So cut it here. Stick the 5' end onto that branch point, onto that adenine. And also at the same time, hold on to exon 1 and the 5' UTR. You don't want it just like drifting off in the nucleus and never to be found again, because that would be equally terrible. So the spliceosome actually holds everything all together, right? So it holds the removed exon 1 as the Lariat loop is being formed with the intron. And so when that Lariat loop is cut off and removed, right, and you can basically see it kind of falls off here, exon 1 are still in kind of proximity, and now they're stuck together by the ligase activity of these spliceosomes. Right, and so once the intron is removed with these other ribonuclear protein molecules, right, which is a mix of RNA and protein, then this RNA intron gets degraded and those nucleotides get reused. And this has to happen for every single intron, right? All eukaryotic genes have at least one intron. Some of them only have one. Some of them have 10, 12, like huge expanses of non-coding information, which has to be removed before translation can occur. Right, so eukaryotic genes are huge. Eukaryotic transcripts are not. They're similar size to prokaryotic transcripts, give or take. You know, average of between 1 and 3,000 nucleotides long. But the actual gene and the precursor mRNA is like so big, you can't even see my hands on the screen, right? Because all that stuff all those introns have to get removed first. Anybody got questions about this stuff? It's kind of a to take on board. Just as long as you remember like the purpose, why this has to happen, consequences of it not happening, and then the basic steps of cut, fold, cut, stick, basically. Okay, I'm not sure we're going to get time to finish this in the last few minutes. Maybe, maybe not. So anyway, all that's happening, RNA polymerase is going along, transcribing the gene. The kappa's been added on the 5' end. Introns are being removed. Now the transcript is cleaved, right? And it's typically, you know, a shortish distance downstream from the end of the, essentially where this polyadenylation has to occur. So this precursor mRNA is cut off, right? And the rest of the RNA that's being transcribed is degraded by RAT1 to terminate transcription. And this 3' end is a lovely juicy target for RNases, right? And so before this can be recognized by RNases, we have a enzyme that adds just A's called a polyadenylase. And this tail can be, it's a long time since I actually looked at it in any particular detail, but 30 to 100 A's long, give or take. And this ATA, actually in many ways, I kind of think about this as like a telomere, like a buffer, right? Its role is to serve as a kind of a landing spot for proteins. And they're unimaginatively called poly-A binding proteins. And it's the presence of those proteins that protects the 3' end of their transcript. And so that's brought us to the end of our hour, now allowing me to go get that second cup of coffee. Yay! Love coffee. And we'll come back in 15 minutes and we'll just finish up this, kind of sum up what that's going to look like. And then we'll get started on translation. All right. So go stretch your legs, grab yourself a cup of tea or coffee or something, and I'll see you in 15 minutes. Bye.